Madame Cécile Delacour, nos va a presentar algo sobre mirando la actividad neuronal a través de múltiples escalas. Me imagino que es un trabajo muy interesante. Le damos la palabra. Muchas gracias.
Okay, so hello everyone. So thank you for this event, despite the complex situation, and thank you for inviting me to give that talk.
Thank you for participating.
So
yeah, we have only 1 minute and a half. Yes.
Yeah. So our goal is to picture neuronal activity, and so to do so, we have 4 main challenges to tackle. First, we are developing a highly sensitive array of nanosensor, basically silicon nanowire and graphene field effect transistor. And second, we look at the biocompatibility of the material we use for neural interfaces, as well as functionalization strategy to improve long-lasting recording and to add new functionalities, such as drug or neuronal growth factor, for instance. So the last point we develop is to realize a model architecture of neural network in vitro in order to model computational processing or disease. So the first task regarding the nanosensors has been motivated by the need to reduce the electrode size for increasing the spatial resolution and the need of more sensitive device to catch as many signals as possible. So here I have compared conventional microelectrodes commercially available that are here in black and silicon nanowire field effect transistor. So we can see on that microelectrode we can see here that microelectrodes are really huge compared to neurons, and it's basically very difficult in that case to record signal propagating to this complex noetic architecture. At the opposite, silicon nanowire are well smaller than the right and can easily be implemented along a single cell and its network in order to track spike propagation. So we realize dense array of those sensors, around 100 devices per chip, and then we culture hippocampal neuron on those chips during 3 weeks until the network becomes mature. So here neurites have been guided above the sensor array, and we can see here the 2 trace of, so the time trace record from 2 adjacent nanowire that show typical response of neural spike. So these 2 signals are delayed by few microseconds that correspond well to the propagation time of a spike from 1 to the second electrode location. So here's basically the same experiment, but with array of graphene field effect transistor that we have realized on transparent and flexible substrate. So in this case, the network is not organized, but cover all the surface. And again, this is the typical signal that we record here, and you can recognize the typical shape of neural spike. So the typical response of the transistor to neural spike. So both technologies, silicon nanowire and graphene, are mature now. And if we compare the 2, they have both real advantages. Silicon nanowire provide a high spatial resolution with a signal to noise ratio which is comparable to the graphene devices. The advantage of the graphene technology is more regarding its optical transparency, flexibility, and also its extremely high neuronal affinity that I will discuss later during this talk. So silicon nanowire has the highest spatial resolution, but it's not completely impossible to record nanoscale event with graphene field effect transistor. Indeed, we can use a nanoscale defect in graphene, such as a grain boundary. So CVD graphene is polycrystalline. So it means that we have a graphene grain that grows, and when these grain merge together, they form a grain boundary here because the lattice are slightly misoriented. And so this is what is shown here. This is a SEM micrograph taken before the complete growth of a graphene monolayer. So you can see in dark the graphene grain, and in bright the future location of grain boundary. So first, people try to avoid those grain boundary because they reduce the mobility of carrier and they reduce the sensitivity of the device. But a few years ago, it appeared that this defect line is highly reactive and highly sensitive, and it can be used for the detection of nanoscale events. So we use it. We use this one-dimensional defect line. So we put a contact around a graphene boundary, and we apply it to measure the ion channel activity within our neurons membrane. So we can recognize here two conductance states that are associated to the opening and closing of ion channel. And this dynamic is sensitive to neurotoxins such as bicuculin or tetrodotoxin that increase or block the neuronal activity. So this is a really exciting new tool. So, but the remaining challenge is to locate this sensing site, is to control the position of the grain boundary. So now I come to my next point. So regarding the biocompatibility of graphene. So on this optical picture, you can see graphene that has been deposited on glass cover slip. And here the bare substrate, so without anything, without any coating. So this is a control part. And what we observe is that neuron can adhere on the bare graphene without any cell adhesive molecule coating. So the adhesion is higher, but also all the growth process is enhanced. So the differentiation or the neurite spreading. So this positive impact on neurons growth is also observed in vivo. So here we have encapsulated intracortical probe with graphene and inserted it within the motor cortex of a rodent. And typically, when you do so, you have a huge immuno and inflammatory reaction that lead to the proliferation of glial cell around the implant. And what we observe with graphene is that these glial cells, the astrocytes, which are in red, are really reduced compared to the control sample, so without graphene. And the population also of neurons is higher around the implant. So this result as consequence also on the detection efficiency, because the number of working sensors is higher for probe coated with graphene. And what does that mean? It means that the number of working electrodes and the number of spikes detected by the electrode is higher. So this is important because when you did such experiments, you want a maximum efficiency and a stable recording in time, because we cannot did it again. It's too invasive. So now this is my third point regarding functionalization strategy. So we have worked on several approach. And here I show you recent work that combine hyaluronic acid, which is a natural polymer present in the brain. And you can observe on this immunostaining assay that all neuronal growth markers are widely enhanced thanks to this advanced coating that we can put on any surface, basically. And here we have, all the neuronal markers that are enhanced, such as the neuronal density, the synapses, the axon, and the neuritic outgrowth. So those tests was also validated in vivo, and this will enable to improve the bioacceptance, for instance, of rigid silicon technology. So to finish, I have two more slides. I don't know what time I have left, but just two more slides on the work we did about the realization of neural network in order to model computational processes.
You have 1 minute.
Okay,
Please.
thank you. in order to model computational processing on chip. So there are many pioneer experiments, but basically here what people do, what people show is that there is a strong, very strong relationship between the organization of the network and its function. So based on this framework, we have cultured neurons within this microfluidic chamber. And depending on the stimulation and on the geometry of the network, we measure the propagation of spike and study learning processes in ills or disease condition. So those fluidic chamber that contains the cell and those micro channel here are really a new key for the realization and for the investigation of model network, because it offers an optimal control of the network geometry during several rakes. So, okay, so to conclude, this is just a few take-home message. So the technology of silicon and graphene field effect transistor is ready to be applied on large scale for studying neurons network. The fabrication is reliable and the sensitivity is high enough to detect single spike and to monitor ion channel activity. The biocompatibility of graphene and the functionalization strategy that we develop opens the way for chronic recording in vitro and in vivo. And finally, we did a huge step in the development of neurons network in vitro with user-defined geometry that are now highly stable in time, meaning that we can keep cell confined in a certain geometry during several weeks. Okay, so our next move will be to combine all this technology with optical indicator, such as thermal sensor, for instance. So this is a work that we have started with Jardin Dantel, so a colleague at Nel Institute. So these indicators are here along the neurites, and we look at the fluorescence, and it will give us an access to study local variation of temperature within neural network. So I want to thank
I think we need
PhD
to,
student and colleague. We work on that project, and thank you for your attention.